In our experiment, we investigated if samples of water with different sized polystyrene beads could be diffusing at different rates. This could be important to understanding how diffusion works in general. It could also give us insight on which size beads will diffuse quicker. The goal of our experiment was to test velocity of various beads across the same amount of time and average the velocity of all of our data points for each size sample to relate back to rate of diffusion. Our data finds that when comparing the average velocity of 5 micron beads and 2 micron beads, that the 5 micron beads diffuse at a higher velocity. These findings are the opposite of what we expected, since we expected the smaller size beads to diffuse at a higher velocity.
Certain parts of our experiment were added in order to reduce human errors. Overall, we just needed to capture videos of the beads diffusing after being squirted onto the clean slide. Using the proper setup, the video was able to be converted onto Tracker, where most of our analysis began.
Our experiment consisted of 7 steps. In step number 3, we made sure to avoid forcefully squirting the solution in order to avoid human error, which would result in the motion being a direct result of the squirting, not diffusion. We made sure to use the same person each time to perform the squirting part of our experiment for consistency, and ensure we were holding the pipette straight up and down every time for precision. The clarity of the microscope and our viewing method could be improved in future experiments.
The motion of each bead with respective sizes after they were squirted onto the slide under the microscope. It is hard to see the difference in velocities with these videos, so we'll break down the differences graphically on the next slide.
The graph on the left represents the position of each 5 micrometer bead tracked over time, and the graph on the right represents the same information collected for the 2 micrometer bead. Both are graphed within the same domain, and as you can see, the 2 micrometer beads yield more consistent results, whereas the 5 micrometer bead had a little bit more inconsistency with greater periods of change in position.
See by the intervals of these displacement graphs, the 5 micrometer beads had a larger total displacement compared to the 2 micrometer beads. The majority of the 5 micrometers had a total displacement larger than the 2 micrometer beads.
In order to draw on our knowledge of physics, we focused on how the velocity of the beads were altered by a change in bead size. The graph shows that the 5 micrometer beads in blue had a larger average velocity than the 2 micrometer beads in orange. The inconsistency of the 5 micrometer data collected is evident when compared to the 2 micrometer beads. This data leads us to believe that during this experiment, the 5 micrometer beads moved at a faster speed and diffused faster into areas of lower concentration. This experiment, force was held constant by pipetting the solution onto the plate consistently. Since force was constant and size was the independent variable for this experiment, Newton's second law helps explain why the bead with a larger mass maintained a larger average velocity. The larger mass of the 5 micrometer bead had smaller acceleration to slow it down when compared to the 2 micrometer bead, which is proved by Newton's second law, rearranged in terms of acceleration. Acceleration equals force divided by mass, so that acceleration is inversely proportional to force. This means that when the 5 micrometer bead was squirted onto the slide, they maintained the larger velocity coming out of the pipette more easily than the 2 micrometer beads. Overall, the force of the pipetting, squirting onto the beads onto the plate, influenced the rate of diffusion by making it so that the 5 micrometer beads exhibited greater momentum after being squirted onto the slide.
Here is a computational model of the 2 different bead sizes colliding with each other for reference.
Force remained constant during each trial. The acceleration of each diffusion event is what changed according to the respective bead size. The data makes sense because the 5 micrometer beads take up more physical space in the slide and are going to collide more often with each other because they are more crowded in the solution. In comparison, the smaller size of the 2 micrometer beads will cause for less collisions, which makes them diffuse at a slower rate. The data that we collected was the opposite of what we hypothesized. Initially, we thought the larger beads would diffuse at a slower rate because of how mass affects velocity in the equation for momentum.